So we welcome Dr. Pengyi from University of Sydney to give his keynote address. And he is also an investigator at the Children's Medical Research Institute in Sydney. So he has a lot of experience in analyzing different types of omics data, what he calls trans-omics analysis, and he also runs a wet lab. So this is a real systems biology approach, and we are looking forward to Pengyi's talk. So Dr. Pengyi, without any further ado, I'd like you to get started. Thank you.
Thank you, Shobha. Let me share my screen. Can everyone see my presentation mode?
Yes.
Okay. Good afternoon, everyone. And firstly, I would like to thank the organizer for giving me the opportunity to present my work today. I'm going to talk about computational methods for trans-omics and single-cell omics. Let me start with the big picture. We like to think the cell is a machinery, and within this machinery, we have the core regulatory circuitry of this machinery controlled by what we call trans-regulatory networks, comprised of signaling, transcriptional and translational and epigenetic regulations. And our research goal is to understand trans-regulatory networks cut across multiple regulatory programs in controlling cell identities and cell fate decisions. So today I'm going to talk about two aspects. The first one is trans-omic analysis of pluripotency transition in embryonic stem cells. And the second one is about computational methods development for analyzing single-cell omics. I will start with the first aspect, which comprised of two recent studies we performed. So we know zygote can expand into a blastocyst, and eventually this can develop into an animal. And from the inner cell mass region of the blastocyst, we can derive embryonic stem cells. And when cultured into a cultured media, those cells are known to be in the so-called naive state. And we can push embryonic stem cell into epiblast-like cells, correspond to the so-called formative state, and that's primed for differentiation. So during this early differentiation process, what we did in the first study is to profile different omic layers to understand this transition process. What we did is to use mass spectrometry to profile the proteomics and the phosphoproteomics, and we use the next generation sequencing technique to profile the transcriptomics and epigenetics. So before we get into the data, the first question to ask is whether the experiment worked as what we expect. So what we have here is bright field images. We have embryonic stem cell in a 2I culture media, and they have this round colony shape. And when we induce the cell to differentiate, we see clear morphological change. We perform qPCR to check the key genes. We have NENA, KL4, PRD14s. Those are the master transcription factors that's known to be associated with naive state. And we see those markers downregulated during the differentiation process. We have FGF5, OTX2. Those two markers are known to be associated with the prime state or formative state, and we see the upregulation of the expression. So these markers suggest the experiment worked as what we hoped it to be. So from there, we performed the multi-omics analysis. And just a summary slide here, we have on the phosphoproteomic level, we identify more than 30,000 phosphopeptides, and this corresponds to more than 14 high confidence phosphosites quantified across 12 time points. On the proteomics level, we identify more than 10,000 proteins, and we have more than 9,000 of those quantified across 9 time points. And for the transcriptomics, we have identify more than 15,000 transcripts across 8 time points, and this corresponds to 13 unique genes, 13,000 unique genes. And for the epigenetics, we have different number of genes enriched for different histone modifications as well as Pol2 ChIP-seq. So the data coverage is very high. The next question to ask is, what about the quality of this multi-omic data set we generated? So we're applying data pre-processing and normalization to process each of these omic data type, which I'm just going to hide the details here. And I just present the results. So on the RNA-seq level, on the transcriptomics, we have 2 biological replicates. And we see the biological replicates at each time point are clustered together and separated by time. And we see the same pattern for the proteomics as well as the phosphoproteomics. For the proteomics, we have 4 biological replicates, and the pool sample, we see again the biological replicates as well as the pool sample are clustered within each time point and separated by time. And this is observed on the phosphoproteomic level as well. So these data suggest the reproducibility of our biological replicates is very high. So from there, we want to begin to ask some interesting biological questions. So what we firstly did is to look at the global dynamics. What we used here is basically a principal component analysis approach. So we plotted here the biological replicates from the transcriptome profiling. As you can see, each of these biological replicates at each of these time points are clustered together and separated by this very nice trajectory. We observe the same patterns in proteomics, phosphoproteomics, and different histone modification marks as well. One of the interesting thing I want to bring your attention to is to see the clear change in the early time points in the phosphoproteomics. Even at 5 minutes, we see a clear transition on the phosphoproteomics level. However, we did not see much of a change in the early time points on the transcriptomics as well as the proteomics. So even at 1 hour, the samples from the early time points are clustered very tightly together. So to properly quantify this dynamics, what we did is to feed a curve across the differentially regulated phosphosite or the transcripts or the proteins, and we'll also overlay some of the histone modification marks here. So what we see is the phosphoproteomics have significant change even at 5 minutes. However, on the transcriptome, we did not see much of a change even at 1 hour. So it takes about 3 hours for the signaling to be passed down to the transcriptional level. And on the proteome level, we did not see much of a change even at 3 hours. So it takes about 6 hours for us to see the regulation to be translated into the protein level. For the histone modification marks, we plotted here H3K4 trimethylation and H3K27 trimethylation. The K4 trimethylation is correspond to activation of the genes, and the 27 trimethylation is associated with repression. So what we see is the activation histone modification marks peaks first, and that suggests probably the genes associated with the formative state in the epiblast-like cells are activated first. And then we have the repression of the genes associated with naive state turned off. So there's a clear epigenetic ordering in the regulation as well. So the next thing we investigated is the level of regulation. So what we plotted here is the magnitude of change on the proteomics or the phosphoproteomics or the transcriptome. And what we see is on the phosphoproteomics, we have the biggest magnitude of change compared to the transcriptome, then the proteome. And this is quite meaningful once we observe this pattern. It probably means it's easier for cell to regulate on the phosphoproteome level. So you only need to have a kinase to put a phosphate on another substrate to have the phosphorylation regulation. However, for you to regulate in the cell on the proteome level, you need to go through the transcription, and then you need to generate a new copy of protein. And it takes a lot more for the cell energy-wise to regulate on the proteome level. So we believe this is probably one of the reasons why we see massive change on the phosphoproteomic level. So from here, we can conclude the phosphosignaling is both the first as well as the most dynamically regulated layer. So the next question is to ask, what is the driver in the signaling on phosphorylation? We know kinases are the driver. So the first thing to study is what are the key kinases that are regulated in this transition process. So to address this question, we take the phosphoproteomic datasets we generated in this study, and then we apply a computational tool we developed previously called cluster evaluation, or CLUE. In essence, this tool is a computational approach to guide the best clustering using external information of our time course dataset. And from here, we can identify the enriched kinases that's driving each of these temporal clusters in our phosphoproteomic datasets. So what we observe here is ERK and A6K or RSK, those kinases are probably the key drivers in the initial transition of the pluripotency. And we see mTOR has this upregulation, probably the second wave. And we observe a transient behavior of P38 alpha, and we observe the downregulation of AKT kinase. So now we know the important kinases that's driving the differentiation in this pluripotency transition process. The next thing to identify is the downstream target of those kinases. So we want to identify the novel substrates for those kinases. So what we did is, again, to take the phosphoproteomic dataset and use another computational tool we developed recently. In essence, we have this machine learning approach that integrates the dynamics from the phosphotemporal profile as well as the kinase recognition motif. And then we combine these two information into generating for each kinase the likely substrates they are phosphorylating. And from there, we identify a lot novel substrates that's previously unknown to be regulated by those kinases in ESL differentiation. In particular, we highlighted those master regulators such as DNMT and KDM. Those are the histone demethylase and DNA methyltransferase. And we also have some other transition factors that's also phosphorylated by key kinases. So now the question is, are those transition factors or master regulators involved in the pluripotency transition? And to address that question, what we did is to rank the genes that are highly expressed in embryonic stem cell and downregulated during the differentiation process on the left-hand side. And we also have the reverse. We have the genes that are upregulated in the epiplastic cells in the formative state. And those genes originally have very low expression, so they are on the right-hand side. And just to zoom in, we highlighted here the master regulators, including transition factors, chromatin remodelers. And as you can see, we have multiple KDM proteins as well as DNMT proteins showing up in these two extreme directions. And those are the ones we predicted to be underlying the signaling cascade. So this links the transcriptional regulation together with the signaling cascade. So the natural next question is to identify the target genes of those transition factors and master regulators. And it actually appears to be quite a difficult question when we begin to investigate this. So we know transition factors are cell type specific. For example, transition factor 1 and 2 are expressed in the naive state, and they are not necessarily expressed in the formative state. And even if they are expressed in the formative state, the binding site of the transition factor can change. So it's difficult to identify transition factor target genes when we have all these dynamics happening in the system. And currently, a lot of computational approach to identify the target genes for the transition factor is to based on the chip-seq profile to identify the closest transition start site of a gene and assign that to the binding site of a transition factor. For example, transition factor 1 here has 2 binding sites in this genomic location. And using the nearest gene transition start site assignment approach, we will assign both of these 2 binding sites to this target gene to say this is the target gene of these 2 binding sites. However, we know there are chromatin interactions, and for the distal binding site of those transition factors, they can well regulate other transition start site of the genes through chromatin interactions. So that type of information can be captured by using CHEPA or Hi-C type of data to identify the interactions. But even with the interactions, it does not necessarily mean if we have the interaction, the target gene will be regulated by a distal binding site of a transition factor. So how do we go about to identify the high confidence transition factor target genes? We design a workflow. So what we did in this study is to identify, firstly, transition factors that's regulated dynamically during the differentiation process. And then we also selected the ones that has been profiled using ChIP-seq assay. So we know their genomic binding locations. Then from there, we can generate the putative target genes using the naive computation approach I just described. So we can use, for example, CHEPA data to link the distal binding site of the transition factors to a potential target genes, and we can assign the binding site that's right at the transition start site of a gene as the target gene of that binding site as well. So we can generate the so-called putative candidate genes. So the next step is what we were able to do using the multi-omic dataset we generated in this study. So what we basically did is to look at the temporal profile on the transcriptional and the proteome level. And from there, we filter out the less likely target genes of the transition factor binding sites. And we then will just return the high confidence target genes. So let's look at how this computational approach performs. On the top, we have the naive approach. We have the ChIP-seq profile for those transition factors, and we have 7 transition factors here. We have the first 5 naive transition factors. They have very high expression in embryonic stem cell, and they have a expression going down during the transition process. We have 2 other transition factors. Their expression goes up during the differentiation process. Those are the transition factors we call formative transition factors. So what we did is to take the binding site based on the ChIP-seq for those transition factors, and we just assign the genes with the closest transition start site to those binding site, and we call those the target genes for those transition factors. And we plotted here their expression profile during the differentiation process. And as you can see, there isn't a very clear differentiation profile change for those target genes using this naive approach. So the second approach is to use the CHEPA data that I mentioned to establish the chromatin loops and to identify the distal target genes. So instead of assigning the closest genes to those binding site, we use the CHEPA information for us to identify the target genes. So those are the temporal profile for those transition factor target genes. And it looks like it's a little bit better than those. It appears to have a bit of a shape that mirrors those transition factors, but still this is not very striking. So the last approach is to use the computational approach I just mentioned to integrate the multi-omic datasets we generated. In particular, we integrated the transcriptome data and the proteomic datasets to identify the target genes. And now you can see clearly we have those temporal patterns for those target genes that mirrors the transcription factors themselves. And this is not surprising once we observe this. We know mostly those transition factors are activated for their target genes. So if their expression goes down, their target genes expression goes down as well. If their expression goes up, their target gene expression goes up. So it looks like using this approach to integrate multi-omic datasets is able to identify high confidence target genes. So to validate this result a little bit further, we use those independent datasets that generated from different laboratories. We downloaded the transcriptional profiles for ESLs, either with wild type ESL or with the transition factors here knocked down. And since we make the assumption those transition factors are activated for their target genes, what we expect to see is if we knock down the transition factors, then their target gene expression should go down. So when compared to the wild type ESL, we should see a downregulation for their target genes. So what we have here are four box plots here. So for each of these transcription factors. And the first one is the expression for all genes before and after the knockdown of the transcription factor. And we normalize this to be right in the middle, 0. And then we have the second box, which corresponds to the naive approach. So we just assign the transcription start site of the genes that's closest to the binding site of the transcription factors and call those the target genes. And the third approach is to use a chromatin loop to identify the distal binding target of those transcription factors. And the last box corresponds to the computational approach we developed using the multi-omic datasets we generated in this study. And as you can see, in these cases, we have the last box correspond to significant downregulation compared to the other boxes, suggesting the computational approach we developed is able to identify really high confidence target genes for those transcription factors. And they have shown the biggest downregulation in this perturbation experiment. So from there, we want to investigate those target genes and see whether there is anything interesting on their epigenetic regulations. So what we plotted here is the H3K27 trimethylation mark. And as I mentioned before, this is a repression mark. So the more of the signal you observe from this mark, the more repressed the gene is. So here we plotted during the differentiation process, the histone modification mark here corresponds to the target genes for those transcription factors. The first 5 are the naive TF target genes. The last 2 are the formative TF target genes. And as you can see, during the differentiation process, we have the accumulation of this repression mark in the naive TF target genes, suggesting their chromatin accessibility has reduced. So basically the expression goes down and they have been turned off on the epigenetic level. Interestingly, we didn't see more histone repression mark for the formative TF target genes. They did not appear to have more repression to begin with. So this suggests those formative TF target genes are equally accessible in the naive state. So this is quite interesting, and we want to validate this result a little bit further. So what we did is to take the ataxy experiment generated from either naive state, naive embryonic stem cell, or epiblast-like cells. And we asked the question, what is the chromatin accessibility based on ataxy for the naive transcription factor target genes in the naive state and in the formative state? And what is the chromatin accessibility for the formative TF target genes in the naive state and formative state? So as you can see, in the naive state, for the naive TF target genes, we have very high chromatin accessibility as marked by this ataxy signal. And after the differentiation for the naive embryonic stem cell to the formative state, we see the reduction of the chromatin accessibility for those naive TF target genes. And this mirrors very well with the H3K27 trimethylation mark. So we have accumulation for the repression mark, and then we lose accessibility for those target genes as well here. However, we observe very high chromatin accessibility for the formative TF target genes even in the naive state, and they return very high chromatin accessibility in the formative state. So this again suggests those formative TF target genes are chromatin permissive even in the naive embryonic stem cells. So with this information, we propose the following model. We have the naive TF target genes, their expression is very high, and they have permissive histone modification marks as well as chromatin architecture. However, during the differentiation into the formative state, we have the reduction of the expression for those genes, and they get repressed. They accumulate H3K27 trimethylation mark, and they have a heterochromatin after the differentiation. For the formative TF target genes, the picture is quite different. So even in the naive embryonic stem cell, we do observe permissive chromatin accessibility. Even though the expression is very weak, they do appears to be ready for expression. And during the differentiation, we observe the formative TF target expression goes up, and we see correspondingly their target genes expression also goes up. So this model summarizes the permissive accessibility for the formative TF target genes in the naive embryonic stem cell. So hopefully through these two studies, I give you some idea how we can apply computational approach to analyze multi-omic datasets to reconstruct firstly single learning cascade and then downstream transcriptional regulations, and in this case in the embryonic stem cell differentiation process. So now I'm going to just move on to the second part of my presentation. So the first two studies I presented performed on the cell population level. However, we know cell are heterogeneous, and the cell population profiling study we typically perform do not offer that level of information to study heterogeneity of the cells. So we only get to see the average of the cell population. However, with the recent development in single cell omics, we are now able to profile especially the transcriptome of individual cells. So now one of the research direction in our group is to develop computational approach that can harness this level of extra information to understand the cell system. So today I'm going to talk about only one tool we have developed recently. It's called AC-Classify, and it is a hierarchical approach to perform cell classification. So let me, before I get into this algorithm, let me first introduce what is the typical approach to perform cell classification or cell identification. Typically, when we generate a single cell RNA-seq datasets, we want to know what are the cell types in the dataset. So the typical approach is to perform some kind of a clustering. So we generate different clusters, and then we go to each of these clusters and we ask what are the marker genes expressing each of this cluster, and based on the prior knowledge of those marker genes, we try to infer what are the cell types present in the dataset we have generated here. So the difficulty appears to be, what if we don't know any markers prior? And what if we don't have very clear clusters? So how do we determine what are the clusters in the dataset, and how do we determine what are the cell types in the dataset? So now what we did in this computational framework is to develop a computational approach to bypass this clustering and prior knowledge-based annotation step by using a reference dataset. So what we mean by reference dataset is, for example, there are a lot of large-scale atlas datasets generated by different consortium already. So we can take advantage of those datasets to train a model to learn the transcriptional profile for each of the cell type that has been profiled in those consortium atlas dataset. And then we can construct a cell type hierarchical tree. And then building a classification model on each of those tree nodes, we can then take this query dataset and be able to figure out what are the likely cell types in the query dataset automatically without using any markers. So this is a computational approach we have published very recently. It's called AC-Classify. So let me just introduce a little bit about some of the mechanism in this AC-Classify. So what we have, let me just jump back. What we have is on each of these nodes of the hierarchical tree, we have a classification model. And what we do is we select differentially expressed genes or some other type of characteristic of the genes that potentially can distinguish the cell type. And then we use different similarity matrix to rank figure out what's the likely cell type for our query datasets based on those genes. And then we generate a bunch of classification models. And then we took the average or the weighted average of those classification model to generate a consensus to help us to generate a final prediction for the possible cell type for each of these query cells in the query dataset based on our reference dataset. So one of the question is, what if the cell type we're trying to classify in the current data set is not present in the reference data set? The correct behavior for the algorithm is to generate those cells as unassigned. So we shouldn't assign those cells to any of the cell type because we don't have that present in the reference data set. The second issue is, what if we have very small population of cell, very small number of cells in certain cell type? Then the most desired behavior for the algorithm is to not to classify that cells, the query cell to a very small cell type, but just to assign this into a so-called intermediate level. So for example, if a cell can be classified, sorry, I just have some pool showing up on my screen. So for example, if we have some of those very small cell type here, and we do not have very representative training set to capture those cell types, then if we don't have very high confidence to classify to those very small cell types, we should classify those query cells just to the intermediate stage. They will be meaningful because they will tell roughly where those cell type, roughly what the cell type will be for those intermediate cells, but it doesn't force those cells to be classified into the terminal node with very low confidence. So now, after introducing this algorithm, the question is really whether this is actually working. So what we did is to curate what we call the data collections. So we have the pancreas data collections, and we have the other data collection from blood called PBMC. So those data collections contains individual data set that has been generated by different people using different protocols, and it has different cell types and different cells as well. And for the PBMC, we have the same sample, but it's generated using different sequencing protocols. So the question really is, if we use one set to train a model, can we classify the cell types from another data set generated, for example, by different people or using different protocol? So since we have 6 data sets here for the pancreas, and we have 7 data sets for PBMC, we can generate 30 pairs of training and testing combinations for the first data collection, and we can generate 42 pairs of training and test combinations for the second data set. So this is a quick summary of the result. We are definitely not the first one to develop this computational approach to classify cells. There are actually a long list of other alternative methods that has been introduced to do similar task. So what we observe here is to use this PBMC dataset. We saw the classification accuracy using our computational approach performs favorably compared to alternative methods. And this is also observed using the pancreas datasets. So we have a competitive performance compared to the alternative methods based on the benchmark studies here. The other aspect of the AC classify is we allow the user to use multiple reference data sets. So one of the questions I mentioned is that what if the cell type in the query data set is not captured in the reference data set? So in that case, we want to combine multiple reference data set that capture more or less similar cell types. And that not only helps us to cover more cell types, but also helps us to improve the number of cells in each of the cell type, so we can have more accurate classification from our model. So AC Classify can allow user to profile multiple reference data sets and generate consensus prediction on a query data set. So we tested this using the PBMC data set. So I mentioned this data set contains the same sample generated using different protocols. So what we can do is, for example, if we want to classify a cell-seq data set here using each of these other data sets generated using different protocols, this will be the classification accuracy by using other data sets to classify the cell-seq. So as you can see, those individual points here are the model trained using each of these other data sets generated by different protocol to classify cell-seq. And this red dot here is actually to combine all the other data sets as the reference jointly and classify this cell-seq data sets using all the other data set together. So in most cases, we have improved classification accuracy, and that's what we are happy to see. The second thing is, as I mentioned, we have the unassigned and the intermediate cells, and this can happen if we don't have the cell type that's present in the reference data set, and we're trying to classify that cell in the current data set, so we couldn't classify that cell into any of the cell types, so we have the unassigned situation. And for the intermediate, is because in many cases, we don't have enough cells in small cell type, so we don't have very high confidence classification. We couldn't classify that properly, so we classified them in the middle. So by combining all the other data sets to train the model and classify each of these individual data sets, we were able to reduce the number of cells that's classified as unassigned or intermediate. So this is another encouraging result. So not only we can improve the classification accuracy, but also we can reduce the number of cells that couldn't be assigned or couldn't be classified accurately. So this is now a package we provided online. It's on GitHub, but it also can be used from Bioconductor. And for anyone who is interested in this package, please visit the website. And if you have any questions, you can email us, and we are happy to see you apply this package to your data set. So lastly, I'd just like to acknowledge my collaborators from different institutes, and I'm happy to answer any questions that you may have. Thank you.
Thank you, Pengyi. And it was an excellent talk. And as you can see, a very large number of questions are already gathering on the chat box. So the first question was from me. You had a picture on the quantification dynamics early on with the phosphoproteomics, proteomics, transcriptomics picture, and the chromatin changes, the chromatin repression. They looked like phosphoproteomics could be reflecting your HEK3 repression. So would you like to look at that and let me know how you go?
Okay, so I think that's one of the things we didn't really look at. So I think that will be an interesting investigation to follow. So definitely the single learning is from this analysis, the single learning is instructing the transcriptional regulation, and the link from the single learning to the epigenetic regulation is of interest for us. Okay,
thank you.
Thank you.
And the next question is here. Do you want to just look at your chat box? It says there are few available hierarchical clustering approaches, such as UMAP and TSNE. Plus, I know that UMAP is better than TSNE. So may I ask why you choose TSNE instead of UMAP?
Oh, so the UMAP and TSNE is basically the visualization approach to show the data sets. Normally we just apply both of them, and we will see which one actually shows a more meaningful representation for the data set we have in hand. So there's no particular reason in this case to choose TSNE than UMAP. So it just happened to be in our analysis we use TSNE, and the result is reasonable.
Okay, so perhaps somebody can try your approach with UMAP and then they can have a new paper. So that could be another thing to do. So the next question is, the multi-omics data that you have used in your project, is it public data or is it private? If not public, would you be willing to share it on a public domain?
The data set is published already and it's public, and we made a GitHub repository to host the process data. And if you need the raw data, it's also uploaded in NCBI GEO database, as well as the Pride proteomics database. So yeah, the short answer is it's available.
Yes. That's fantastic, because I think you are supporting open access at the limit of what can be done as open access. Okay, then another question. Can a cell be simultaneously classified into two types? And how can this be resolved then?
Yeah, so I think that's a very good question. So in our setting, the cell couldn't be classified into two cell types, but I think the closest we have is assign a cell into the intermediate stage, and that actually then branch into multiple cell types. So you can interpret as being classified into either two cell types, or is actually the progenitor of that two cell types. We do know in certain disease assistance, such as in cancer, some cells may acquire different phenotypes that resemble multiple cell types. So that will be an interesting research direction to actually take. So thanks for that question.
Okay, and then one last question is there. The number of cell types are different in different data sets of PBMC. How to decide it in your methods, SC classify? Thank you.
Yeah, that's a very good question. We actually have one of this framework that I don't have time to go through. So
you do have the time, actually. You have another 4 minutes. If you want to go through, there is no problem.
Okay. Yeah. So we have this evaluation framework because it's a very difficult. It's a very complex setting. So what we have is for predicting a cell type, we first ask whether the cell is in the reference data set. If it's not in the reference data set, then we ask whether is it classified that cell. If it's classified, then that's incorrectly assigned. So in this case, we shouldn't assign a cell. So it has to be unassigned, and that's what we consider as correct. If the cell is in the reference data set, then we go this way. Then we ask whether the cell has been classified. If it's not classified, then to the terminal node, we ask whether it's in the intermediate. If it's not in the intermediate, then that basically means it's unassigned. And in that case, it's wrong. So we shouldn't unassign a cell. It should be at least classified to the intermediate. So if it's classified as intermediate, then we ask the question whether it's on the right branch of the cell type hierarchical tree. And if it's on the right branch, then we say this is a correct intermediate classification. Otherwise, it's incorrectly classified intermediate cell. So the last scenario is whether it's assigned to the terminal cell type, not in the intermediate. If it's in the terminal cell type, then we basically just ask whether the cell is correctly classified or misclassified. So that's how we use, that's how we calculate the classification accuracy in our evaluation model.
So it remains for me to thank Pengyi for a very, very informative talk on 2 different topics, a host of new methodologies, lots of data for everybody to start analysis and new approaches. So thank you very much.